Okay, let's talk protein synthesis. So protein synthesis. We got to make a bunch of different proteins in our body. Like I said before, collagen. We've got keratin. You've got insulin. You got hemoglobin. You got all of your every single enzyme in your body is made of protein. So that protein that we have, let's draw a protein here. Looks like this, like a little globby thing that's all folded up in a specific way. So let's say this is lactase. It's going to break down lactose. And the reason it can do it is because of its three-dimensional structure. It allows it to do that. What determines this three-dimensional structure? The primary structure, which is strictly just because what are proteins made out of? They're big macromolecules and they're made of monomers. What are the monomers? Amino acids. We have 20 different types of flavors of amino acids, and they're all put together in a specific order, right? They're in a specific order here. So this lactase enzyme is always going to be something like methionine, arginine, lysine, proline, valine, etc. Same thing if you've got a hemoglobin. It's always going to be in that same order, then that's going to determine how that structure is made. So where are proteins made in the body or in the cells? They are made in the ribosomes. Remember, the ribosomes are the factories that are going to put together these amino acids, like a popcorn on a string. How does the factory know that it needs to arrange them in this appropriate order? It's going to get those instructions from DNA. It's going to get those instructions from DNA. That's the basic process of protein synthesis. Now there's some steps that we got to put in. So protein synthesis is going to go DNA to mRNA to protein. Transcription is DNA from mRNA, translation is mRNA to protein. If that confuses you, that's okay. So let's look at our cell here. Let's look at our cell. So we've got our nucleus, where our DNA lives. We got our endoplasmic reticulum right here. It's got all those ribosomes that are going to be the factories. And it's got the Golgi complex. It's going to package and ship it, right? Okay, so in here is the DNA. Out here is where it needs to get made, right? In the cytoplasm. DNA is in the nucleus. This is out in the cytoplasm. Your DNA is pretty important. Would you want to take it out of the body? Would you want to take it out of the nucleus and maybe risk getting it damaged? No, you do not do that. If you go to the library and you have to go look at a reference book, maybe they've only got one of them. Now, can you check that reference book out of the library? No, you can't. What do you got to do with it? Say you just want one little section, and you don't need the whole book. How do you do that? You're going to copy that section. Take it and put it on a photocopier. Boop. Copy that recipe out of there. It's a recipe book that the library doesn't want you to take out. Because that's what the DNA is. DNA is the recipe book for making all of your 20,000 proteins that are in your body. So, well, if we can't take the DNA out to the ribosome to tell it how to make our proteins, to give it the instructions to make our casein protein, how do we get it there? How does it get the information? Well, like I said, you make a photocopy. The photocopy of DNA, which is just going to be a small section, because your DNA isn't just coding, like the whole thing isn't just coding, only small sections of it. Those are called genes. The small sections of DNA that are going to code for a protein. So what we do is we have a thing called mRNA, which is messenger RNA. Going to send the message to the ribosome. So let's assume that in the nucleus, we like our ribosome is like, hey, we need to make more lactase. Our person just drank a ton of milk. All right, cool. So inside the nucleus, they get the message. They've got to get the recipe out to the ribosome. So how are they going to do that? mRNA is going to come in and it's going to copy a short segment of the DNA. It's going to take the instructions. It's going to take that out to the ribosome and it's going to plug it into the ribosome, and the ribosome is going to read it and go, okay, cool, I got the recipe. Let me put it together into the appropriate amino acids. There's two words for this. There's transcription and there's translation. Transcribing means just writing stuff down. You're copying it in the same language. When you have a transcriptionist service, like I call up and I say, okay, the patient's 46-year-old male, has complaints of low back pain. Somebody will write a report on that. They transcribe it. Still the same language, It's just a little bit different. So DNA and mRNA, they're nucleotide language. You're just transcribing. Where does this take place? Where does transcription take place? Inside the nucleus. So you're going to have an mRNA polymerase. It's going to go in, it's going to find a section of DNA, go, oh, here's the recipe. Let me make a copy of it. Then the mRNA is going to leave the nucleus. And then what it's going to do is it's going to get read by the ribosome, and the ribosome is going to translate from nucleotide language into amino acid language. That is translation. Where does that happen? In the cytoplasm. Our cell needs to make protein. The protein is made in the ribosome. The ribosome is in the cytoplasm. The ribosome needs information or instructions on how to appropriately put the amino acids together to get a functional protein. That information is housed in the library, in the nucleus. We can't take our DNA out of the nucleus to bring it to the ribosome. So what do we have to do? We got to make a photocopy of it. What is that photocopy called? That is mRNA. mRNA, going from DNA to mRNA, same language, nucleotide language, you're just transcribing, you're just writing stuff down. You take the mRNA language, and you take that mRNA, and it goes out into the cytoplasm, you bring it out to the ribosome reads it, and translates it into the language of amino acids, from nucleotide language to amino acid language. That is what makes our protein. Then it goes to the Golgi complex, where it gets exocytosed out into the extracellular fluid. The rules are the same with this. This is where we learn the law of complementary base pairs. So C to G, T to A, but anywhere we got an A, we got to put a U in. All right, just to look real quick. I just want to look right here. All right, there's 3 other words we gotta know. We have to know what a base triplet is. We got to know what a codon is, and you got to know what an anticodon is. DNA language is in sets of 3. This is like Morse code. Every 3 sets of letters will eventually represent an amino acid. So each 3 of these, that's why they're called base triplets. Now, this is in the nucleus, right? RNA polymerase is going to come in. It's found our section to make our protein. Here's the protein that we're trying to make. This is a gene. That's the section that's going to code for that. mRNA comes in. It sees an A. What's it going to do? Or it sees a T. It's going to put an A. It sees an A. It's going to put a U. Puts a C. It's going to put a G. That is what a codon is. That mRNA is going to go out to the ribosome. How does it know where to start? AUG is what is called a start codon. There are also stop codons. Stop codons, like UGA. There are a few other stop codons. So when it gets to this part, the ribosome goes, oh, good, we're done. AUG is the start, because remember, this is going to go into the ribosome. It's going to read AUG, and that's going to be the start. And so it's going to build this amino acid until it gets to UAG, and then it's going to stop. What is an anticodon? We have this structure called tRNA. tRNA is essentially, it holds on to the amino acids in the cytosol. So it's basically like holding a raffle card right here. And so when the ribosome reads the start codon, it reads it like it's reading a raffle ticket. It goes, okay, everybody, pay attention inside the cell here. We're going to read our first codon off of our mRNA. Is everybody paying attention? All you tRNAs, you're paying attention? Make sure you look at your code to see who's the winner. And it says AUG. And all the tRNAs, they look at their anticodons, and the one that has the complementary base is the winner. So AUG, the complementary base, is going to be UAC. I'm the winner! I come down here, and I give my methionine to the ribosome. Awesome. Next one, GCG. This tRNA looks down and goes, oh, see, GC. I've got the anticodon to their codon. I am the winner. I come down, and I add my amino acid onto there. and so on until you hit the stop codon. Okay, so what do you need to know? What is translation? What is transcription? What's a codon? What's a base triplet? What's an anticodon? What's a start codon? What is an example of a start codon? What is an example of a stop codon?